So remember the H plus, so it gets the nickname proton. So these guys are electron transporters, so they're going to transport electrons, but they're going to do some work. So I stood on a lab bench for you guys before, talking about electrons, remember that? So what did we say? Why was I doing that? What was I telling you about electrons? Why did we go from one layer to the next? So what is it? What's so important about that?
The electron, they release energy as they jump.
Energy. Yeah. There's energy in electrons. Well, what is energy used to do? Work. So these guys are going to get electrons, and they're going to use the energy electrons to do work. Their work is to take a proton and pump it to the intramembrane space. Now let's see why that would be important. Outer membrane. Did it go out there into the cytoplasm?
No.
Did it come to the matrix?
No.
So when they pop the hydrogen ion into the intramembrane space, what's that?
Stays there.
Stay there. Say that again. Somebody said it.
Trapped.
It's trapped. It is trapped. Now think Hoover Dam. What do we use Hoover Dam for?
To trap the water.
To trap water. And then after we trap the water, we can put these big things called generators, these little turbines.
And they move them.
And it turns, and what are those turbines producing?
Electricity.
Electricity. We can't dam up water in cells. Water goes where it wants. But cells can dam up hydrogen. Now here's the problem. Where is all that hydrogen being captured from?
From the
glycolysis. Where else?
Wherever where you have NADH to transport it. So Krebs cycle and glycolysis.
Okay. You're an NAD molecule. You're an NAD molecule. You're an NAD molecule. Huh? Harder to come down and get the bag. Alright, so NAD. What do you do at the Krebs cycle? What do you do over at the Krebs cycle and glycolysis?
You pick up hydrogens and shake them to the electrons.
Strip hydrogen.
So when they strip hydrogen off, they go from NAD to NADH. So you just got, what is happening to him? He's oxidized, so now he's getting reduced.
Why did you get 2 electrons?
We'll talk about it in a second,
Okay.
Gabriel. All right, does the NADH need to stay there? We're going to cash it in?
No.
You got to travel to the bank. Think about it. When you make your check at work, where do you got to travel? To the bank. So you guys have got to get travel. Huh?
Come on, Carter. Shake it.
Well, travel. Bring your electrons and hydrogen because you've got to cash them in. Alright, so let's talk about the electron transport chain. There are a lot of proteins. We're not going to learn all the individual names. If you're in biochemistry class, you learn every individual's name and everything they do. But for the most part, they work in complexes, so we're going to learn them in complexes at this time. So you've got a complex 1. It is going to be the one that's going to receive the electrons from NADH coming in. Complex 2, we're going to have there's a part of this complex, there's another little protein in here that's in the membrane that will shuttle from complex 1 to complex 2 and carry the electrons over, and they get deposited. And then those electrons, the energy, are used to do work. And then we have another one that shuttles to the 3rd complex. Now, I know these sound like silly questions. You shouldn't have anything in your hands. Hand those back. We got to set some ground rules first. How many hands do you have, Gracie?
I have 2.
Those are no longer hands.
Okay.
That's an electron slot, and that's an electron slot. Lane, how many do you have?
No. 2 electron slots.
2 electron slots. What you got, Stephanie?
2 electron slots.
Those are electron slots. They're not hands anymore. Y'all hear that? So how many, Gracie, how many electrons can you hold at a time?
2.
Just 2. No more than 2. So Blaine tries to hand you some. What are you going to do?
Throw one away.
No, you can't throw nothing away. Nobody gets to throw electrons away. That's dangerous.
I need it? I don't know.
No.
Oh, you give it to the oxygen.
Take some water.
You pulled it.
It goes back to
You're right.
That's
You're right
why
too.
That's why it's called
Combine them?
an electron.
No, you
yeah.
can't do anything until somebody comes and takes them from you.
Okay.
Alright, so NADH comes to the first complex, and NADH hands the electrons off first. And then the energy in those electrons are going to be used to take the hydrogen and pump it to the intramembrane space. Just toss it back there.
So why did you take 2 electrons? That's what I want to know.
Alright, so
the
both of your
hydrogen
electron
had one.
carriers carry electrons in pairs. So you'll actually, in a biochemistry book, higher level books, you'd see it be written as NADH plus H. And FADH is written as FADH2. So they actually carry in pairs. We're just going to say NADH is carrying hydrogen.
Okay. So
we're
it's
gonna
carrying
say
2 molecules instead of 1.
it's carrying 2 electrons, carries in pairs.
Okay.
Now, so NAD has dropped off. What form is it in now?
And
Oxidized.
yeah.
Now, oxidative phosphorylation is a term you guys are going to learn. What did he do to this protein complex when he passed off electrons? Reduced it, and he became oxidized. Pass your electrons. What just happened to this complex?
Oxidized.
Reduced.
Reduced, while this one became.
Oxidized.
You're going to use the energy to pump that proton. Then we pass the electrons again. This one's going to get
Where did she
Oh,
get
that
the
was-
proton from?
from the matrix.
Oh, so she gave it to her to
No,
float?
they pull it from the matrix.
They just chill.
They're floating around right here.
Hydrogen ions are available here. Pumps. Now, Carter goes back to the matrix, and as soon as he gets back to the matrix, just pretend like I gave you some electrons. Go back to line. I got tired of carrying the marbles around. Oh, you thought you were free. That is exactly the way it happens in the cell. That idiot is like, I am free, I am unloaded, and it's like, come here to the citric acid cycle. As soon as it gets back, it's going to get loaded. Because what do we need to constantly be making a supply of? ATP. Those things don't sit empty very long. So the next one comes in, drops off. So you're going to get oxidized NAD, and then you're going to get reduced again, pump it, and then pass, and then pump. Sorry, hold up. This is how y'all learn. Pump it. All right, so we got to think about something for a minute. I want you to process. So all of these carriers have already dropped off their loads. They've already been reduced again, and look where they are. Back in line.
So this is a continued cycle.
The continuous cycle, yes. But
down here.
now let's think about this for a minute.
But they're
Can
all
those
full.
guys drop off?
No.
All right, so think about that. You guys are running the citric acid cycle. Some of you are those intermediates. Some of you guys are the enzymes doing the work. Can you run the citric acid cycle right now? Why not?
Because we don't have NAD to come in.
All
and
your
pick.
trucks are loaded, aren't they? They're loaded and gone. So if you go to that rock truck crusher analogy, the crusher can't load any trucks if they're all gone.
It's just like if one process is stopped, then it just stops the entire process.
Now, that stopped the citric acid cycle. Let's go out here. There was NAD, reduced NADH out here.
Not letting them in anymore.
Now, it's going to come in, but here's the problem. Let's see who remembers the glycolysis. Could you make ATP before stripping hydrogens off?
Yes, 2.
No,
No.
you used them.
You had to take the hydrogen off to be able to put the phosphate on to start making ATP. So what's happening in glycolysis right now?
Oh, it's also stopped.
It's also stopping. So how you feeling right now?
It's sluggish.
You're dying.
Tired's right, I guess so.
So we got a problem here. You said we needed what to come here?
Oxygen.
So come be oxygen.
Jeez.
You like to pick on everybody else.
Right.
Alright, so when oxygen comes to the end of the electron transport chain, now think about this. they accepted electrons at glycolysis and citric acid cycle, correct? But were they the last ones to handle those electrons? No. These guys have all accepted those electrons, but are they the last ones to handle them? This person, this substrate is going to be the last one to go away with them. This is called the final electron acceptor. Now here's the problem. You're not only pumping protons and handling electrons, but you are enzyme by nature. So whatever the final electron acceptor is, you've got to be able to recognize them. Remember that active site concept? This one, because we are in the mitochondria, recognizes oxygen. Y'all follow that? So this whole process now becomes oxygen right here. Aerobic. Right here is what makes it aerobic cellular respiration. And if it's an organism that has mitochondria, guess what kind of respiration we're going to run? Aerobic. Oh, we're starting to get some light bulbs. This is good. All right, so the electrons are passed off, and the oxygen is going to take those electrons, but it's also going to take two hydrogens from the matrix, and that's going to become H2O, water. We make water. Y'all see that? Now what can start happening again? They can keep flowing. So we'll go back to the questions at the beginning of class. Why is oxygen so important for you?
Because it runs the electron
respiration
chain
can't happen without it.
Electron transport can't happen without it. What happens to the citric acid cycle?
Stops.
It stops. And if you've got an organism that's strictly aerobic, or a cell that's strictly aerobic, what's going to happen in that cell? It's going to die.
Certain death.
Yeah, certain death.
So it's only an electron transport complex 3 that recognizes oxygen.
That's in an organism that runs aerobic cell respiration. Now let's change everything up. NADs, thank you. Oxygen, thank you. I'm not done with you guys. You can sit down. So now we're going to change the whole scenario. We're going to switch cells entirely. So we've been in the mitochondria this whole time. We're going to pick up and we're going to go be in another organism. You are no longer in the matrix. We are now a bacterial cell. So where is the citric acid cycle going to happen?
In the
Cytoplasm. So where are they standing?
Cytoplasm. Right next to the membrane.
Not right next to. They're actually part
In the membrane.
of the membrane.
Oh, okay.
You're embedded in the membrane. So these are some of those embedded proteins. Now I'm going to get. Sorry, JC.
Weren't they
Take this
embedded
title
in
away
the?
from you, and I'm going to
Weren't they embedded in the mitochondria before, in the membrane of the
in the
mitochondria?
inner membrane, embedded in the inner membrane. But we're in bacteria now, so we're in the cell membrane.
Okay.
Lily, can I pick on you? We are now in a bacterial cell. Lily does not recognize oxygen. This complex recognizes nitrate. So what kind of process we got now? Anaerobic what? Cellular respiration. Is this fermentation? No. How does aerobic cell respiration and anaerobic cellular respiration differ?
Final
Electron acceptor. All right, Lily, I'm gonna kick you out of here. So there's Lily. Gabe's no longer oxygen. He's gonna play an entirely different role. I'm gonna pick on him. Come on.
No, I'm eating.
You dish it to everybody else. Huh? You're gonna enjoy this. All right. So cows get a bad rap. They get a real bad rap. Say that louder. Methane. So where's that methane coming from?
Booty hole.
Oh my god. From the bacteria in their stomach.
Alright, so cows are animals,
I
correct?
like his answer better.
Cows have mitochondria in their cells. So what kind of respiration are cows running? Aerobic. Are they producing some gas?
Carbon dioxide.
Yes,
what are they
CO2.
producing at the end?
Water.
They're making water.
So they produce.
But in the gut of the cow, they have bacteria, and some of those bacteria will have a third complex that doesn't recognize oxygen, it doesn't recognize nitrate, it doesn't recognize nitrite, it doesn't recognize sulfate. You guys hear all the different anaerobic final electron acceptors? This one recognizes carbonate. And when it takes carbonate and it hands those electrons off, that carbonate becomes CH4, methane gas. So it's not the cow producing the gas, it's the cow's bacteria.
And if we get rid of this bacteria from them,
You do that, they can't process their foodstuffs because they're dependent on them to be able to process their foodstuffs in their gut.
Is it the grass that they eat that the bacteria feeds off of, or is it what we feed them? Because we feed them corn products.
Which also
Both.
has starch in it, so it
Both.
doesn't matter.
So,
So it doesn't matter which one the bacteria are going to feed off of, but they are needed to help them break down their food products in their gut. So it's the bacteria.
Isn't it kind of like lactose with us?
Huh?
Lactose and stuff like that with us? A bacteria in our gut that helps process our
They have different ones to help them
Most
process
sugar,
the
I
plants
wait
and
for
stuff they're
them to be
consuming,
on
the
that
plant
black.
products.
but she's saying, isn't it similar?
Huh?
Isn't
Oh,
it-
yeah.
Isn't it similar?
Yeah, they're gonna have to have some enzymes to help process, so when the cows are processing milk, the calves, they have to. They're gonna have bacteria that help them process the lactose. So how does this make sense as far as electron transport so far? Anybody counting ATP yet?
I guess.
Isn't that what we're supposed to be making?
You haven't yet.
But this is the electron transport
You
chain.
need the
Yeah.
ATP synthase.
The last one was
Say
a
that
windmill.
louder.
The ATP synthase.
Who wants to be ATP synthase?
Trent does.
He's strong enough.
Just so you know, it's one of my favorite all-time proteins. Because I've actually got to see it labeled in radioactive labeling and rotate. It's so cool.
Gnarly.
Okay,
You're at
cool.
the end.
Okay.
I want my oxygen one back. JC,
JC.
We need oxygen.
we want our oxygen acceptor back. So we're going back to the mitochondria now. So you're gonna slide down here. Stand right here. In that hand, you're gonna hold the
Is this still the
ADP. Huh?
cow? Is this still the
No, we're
cow?
back at, we're going back to that mitochondria. So we're back in the, so back in your body, mitochondria or in the cow's cell, not the cow's gut. So we're back in the mitochondria. So we've got one that accepts oxygen to pass off the electrons. Now we're gonna look at what happens to all those hydrogens that we pump back there. ATP synthase. What does the name tell you?
It synthesizes
It
ATP.
synthesizes ATP. So it has two binding sites, and it's so there's the active sites: phosphate, ADP. Now here's what has to happen. All of these hydrogens that were pumped back here, they're dammed up. There are slots that are built into this enzyme that are going to cause that enzyme. So as you turn, those two sites come together. So turn, and they actually push this enzyme. So, and it takes 3 of them. So they just come back in. Keep going. They keep coming back into the matrix. And then as it turns, it slams it together, and we go from ADP and phosphate to ATP. Now think about this. I'm gonna have to build something better. I was trying to work in a hurry. Put the magnets in them, and there's not strong enough magnets. I need much better magnets. So I was trying to get them more magnets with holes, but they're too thick. So you can lay them down now because we've got the concept. So what's driving ATP synthase?
The hydrogen.
So think about it for a minute. I know this is an old school movie.
So it's almost like
Little House on the Prairie.
Never heard of it. I'm kidding.
Are you serious?
I've never heard of it, for real.
Alright, so, but I can teach you still, even though you've never seen it. Little House on the Prairie was set back in the 1800s.
Okay.
Daddy worked at a sawmill.
Windmill, okay.
So sawmills, they didn't have power going to them. So how does the saw get powered?
water.
Water. So they had these big wheels outside the sawmills, and the creek would run, and what would the water hit? It hit like a little paddle on it, and as it pushed, the next paddle would come down and get pushed, and it would rotate, and that rotation produced the kinetic energy to power the saw. Y'all see that? The hydrogen being dammed up is going to push the ATP synthase to drive it. So if these guys don't work, then that don't work.
I'm screwed.
Yes, exactly.
Are we done?
Yeah.
Or what did you say? Certain death. Certain death.
Certain.
So dark.
It's the
So they're like the Colorado Just make River, another cell.
Uh-huh.
and then that's Lake Mead, and then he's the Hoover Dam.
There you go.
Okay.
Exactly. That's it.
like those old timey little wheel things.
Now y'all want to see that in action?
Yes.
Y'all have a seat. I'll pull a video up real quick to wrap this all up.